Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway, okay, and its involvement in cancer. Okay, so we're in the process of discussing the PI3 kinase AKT mTOR pathway. So we've discussed that when a PI3 kinase, a type 1 PI3 kinase, is activated, it's going to convert phosphatidylinositol 4,5-bisphosphate molecules in the plasma membrane into phosphatidylinositol 3,4,5 trisphosphate molecules. Okay, now we've discussed that PIP3 here is not a normal component of the phospholipid bilayer. Okay, and that's because there is this phosphatase, the phosphatase intensin homolog PTEN, which is usually involved in removing any PIP3 that you have and breaking it back down to PIP2. Okay, right. Now, when you get that transient appearance of PIP3 molecules then within the plasma membrane before the PTEN has had time to break it down, what happens is two enzymes from the cytoplasm are recruited to the plasma membrane. One is phosphoinositide-dependent kinase 1, PTK1 here, and the other is AKT, or protein kinase B. And the way that they bind to the PI3 is through plextrin homology domains, pH domains, okay, which bind to special phospholipids in the phospholipid bilayer. Okay, phosphoinositide-dependent kinase 1, PTK1, when it gets recruited to the underside of the plasma membrane by PIP2, not only does it relocate, but it also becomes activated, and it is now a serine-threonine kinase, and it now phosphorylates the protein kinase B, or AKT, which is also now in the same location, i.e. attached to the underside of the phospholipid bilayer by attachment to PIP3. Now, it phosphorylates AKT protein kinase B on threonine 308, and that's the first part of the activation of protein kinase B. We've then discussed that the second part of the activation of protein kinase B relies on the phosphorylation of serine 473, and we said that the scientific literature is now leaning towards mTOR2 as the enzyme complex which performs that phosphorylation. So remember, mTOR2 is mTOR, the fundamental enzyme, with loads of accessory proteins stuck on the side which determine what its targets actually are. And it's not known at present how mTOR2 is activated to perform this phosphorylation that is crucial in the activation of protein kinase B. Okay, now what's going to happen is the protein kinase B is going to detach from the PIP3 now and float back into the cytoplasm. Okay, and in the cytoplasm it's going to perform actions that are going to lead to the activation of mTOR1, the other complex that mTOR is involved in, which has very different accessory subunits attached to it. Okay, right. So I think I'll show you mTOR1 first, and then we'll see how protein kinase B is actually going to activate mTOR1. Okay, right. So mTOR1 then. mTOR1 is simpler than mTOR2. It's only got 3 accessory subunits rather than 5 that we had in mTOR2. So at the centre again you have the mammalian target of rapamycin, serine threonine kinase enzyme here, which we'll colour in red. And now, just like I said that if there was one accessory subunit that characterised mTOR2, the one that you should remember if you're not going to remember all the others, it was RICTOR that characterised mTOR2. Okay, in the case of mTOR1, it is RAPTOR, okay, which takes the place of RICTOR. So this is going to be a RAPTOR. Okay, and what does RAPTOR stand for? Well, it stands for the regulatory associated protein of mTOR. So this is the regulatory, that's the R, associated, that's the A, and then protein of mTOR, the P is for protein, and then the TOR is for of mTOR. Okay, right, so let's colour in RAPTOR there in blue. Okay, and then the 2 other subunits that are now going to attach to the mTOR to make the mTOR1 are 2 that we've already seen. So DEPTOR is going to bind again, and also G-beta-L as well. So I'll put these 2 in the same position as we had previously. Okay, so here is DEPTOR here, the DEP domain containing mTOR interacting protein. okay? And then the other one was G-beta-L, the GTPase beta subunit-like protein, also called MLST8. You may see it referred to as MLST8. Okay, right, and that now is the mammalian target of rapamycin complex 1. Okay, so this is what we're trying to activate. When people talk about the PI3 kinase AKT mTOR pathway, the mTOR that they are referring to in that pathway is the mammalian target of rapamycin complex 1, mTOR1, not mTOR2. Okay, right, so how are we going to activate this protein? Well, this enzyme. To activate this enzyme, you need a monomeric G protein. Okay, so the monomeric G protein that is really important in activating mTOR1 is a monomeric G protein called Reb. Okay, now, like all monomeric G proteins, Reb can be in two states, an on state and an off state. Now, in the on state, it will have guanosine triphosphate, GTP, bound to it, and in the off state, sorry, that's in the on state. In the on state, it will have guanosine triphosphate bound to it, and in the off state, it will have guanosine diphosphate bound to it. And the on state, Reb, is what's going to activate the mammalian target of rapamycin complex 1, okay, in mechanisms that still are not particularly well understood. But we know that Reb, this monomeric G protein in the on state, activates mammalian target of rapamycin complex 1. Now, normally there is not any Reb GTP, okay, you do not have any active Reb in the on state, and the reason is that the moment it's turned into the on state, there is another protein complex that is waiting to turn it into the off state, okay? So there is a protein complex that activates the intrinsic GTPase activity of Reb. So Reb is an enzyme that's capable of hydrolyzing the GTP itself, okay? But there is an enzyme complex that is capable of activating Reb to hydrolyze its GTP down to GDP and therefore turn itself off. Okay, now things like that are known as gaps, basically. So we are looking for what is the Reb gap. Okay, the Reb GTPase accelerating protein is what GAP stands for. So the G is for GTPase, the A is for accelerating, and then the P is for protein. Okay, so we want to know which protein is it that interacts with the Reb when it's in the on state and activates the Reb intrinsic GTPase to hydrolyze that GTP down to GDP, and this is what's known as the Reb gap. Okay, the GTPase activating protein or GTPase accelerating protein for Reb. Okay, right. So what is this Reb gap? Well, it's a complex of two proteins that is known as the tuberous sclerosis complex. So let's now study the tuberous sclerosis complex. So the tuberous sclerosis complex consists of two members. It consists of the tuberous sclerosis complex protein 1 and the tuberous sclerosis complex protein 2. So let me show this. So here is the tuberous sclerosis complex protein 1, and here is the tuberous sclerosis complex protein 2. Okay, TSC2 for short. So TSC is for tuberous sclerosis complex. Okay, so the tuberous sclerosis complex consists of these two proteins bound to one another, tuberous sclerosis complex protein 1 and tuberous sclerosis complex protein 2. And basically the tuberous sclerosis complex is now capable of interacting with Reb when it's in the on state and accelerating the intrinsic GTPase activity of Reb so that it turns itself off. Okay, and that's why you don't have that much Reb in the on state normally, because the tuberous sclerosis complex just turns the Reb off the instant it gets turned on. Okay, now if we want Reb GTP to build up in the cytoplasm and to activate our mammalian target of rapamycin complex 1, then we need to inactivate the tuberous sclerosis complex, because if we inactivate this, then we're going to stop this Reb gap activity, and therefore Reb is going to hydrolyze its GTP incredibly slowly compared to how it would hydrolyze it with the tuberous sclerosis complex interacting with it. Okay, and therefore Reb GTP, Reb in the on state, is going to build up, and it will then be able to activate the mammalian target of rapamycin complex 1. And it's this tuberous sclerosis complex that protein kinase B is now going to target. Okay, so our protein kinase B has now fallen off the cell membrane. It's got its 2 phosphate groups bound to it. Okay, one from the phosphoinositide-dependent kinase, okay, and the other from the mTOR2. Okay, here they are. And now it's a serine-threonine kinase itself. It can phosphorylate proteins, and the protein it's going to phosphorylate is this tuberous sclerosis complex protein 2. Okay, so what it's going to do is it's going to phosphorylate tuberous sclerosis complex protein 2, and when it phosphorylates tuberous sclerosis complex protein 2, tuberous sclerosis complex protein 2 now associates with a very different protein than tuberous sclerosis complex protein 1 called 1433. Okay, so I'm drawing this here. Here's that phosphate group that we have now stuck onto the tuberous sclerosis complex protein 2 here, and that was the doing of this protein kinase B enzyme over here. Okay, so here is protein kinase B, or AKT. Okay, and now once you've stuck that phosphate group onto tuberous sclerosis complex protein 2, this new protein here in orange, which is the 1433 protein, has now bound to the phosphorylated tuberous sclerosis complex protein 1, tuberous sclerosis complex protein 2, and stopped the tuberous sclerosis complex protein 2 from interacting with the tuberous sclerosis complex protein 1. Okay, so this now causes the dissembling of the tuberous sclerosis complexes, because now all the tuberous sclerosis complex protein 2s are going to end up bound to 1433 rather than tuberous sclerosis complex 1. And on its own, tuberous sclerosis complex 1 isn't capable of the Reb GTPase accelerating activity. Okay, so that means that now you're not going to be activating the Reb GTP complexes to turn themselves off. Okay, so Reb GTP is going to build up within the cytoplasm, and now it's going to be able to activate the mammalian target of rapamycin complex 1 in ways that are still not completely understood. Okay, so that now is the final component of the PI3 kinase AKT mTOR pathway as far as the name of the pathway is concerned. Okay, we started with the activation of PI3 kinase that led through the generation of PIP3 to the activation of protein kinase B, or AKT, and now we've activated our mammalian target of rapamycin complex 1, which is what the mTOR in the name means. Okay, right. So now, what does mammalian target of rapamycin complex 1 actually do? Well, we talked right at the beginning about what it does. It's going to allow the translation of all of this new mRNA that we're producing because of the changes in transcription in these cells that have been activated to divide. Okay, but how does it actually activate the translation of all of those new mRNAs that we've produced? Well, it has 2 main targets, okay, which I'm going to mention now. So, firstly, one of its targets is that it phosphorylates another kinase enzyme, okay, another serine-threonine kinase enzyme here, okay, and activates this kinase enzyme. Okay, so here is this phosphorylation event here, done by the mammalian target of rapamycin complex 1, now that it's activated. And it's going to activate an enzyme known as S6K, which stands for S6 kinase. Okay, and now once you have phosphorylated S6 kinase, it's now activated and it's going to phosphorylate a protein called S6. Okay, now where is S6? Well, basically S6 is a protein within ribosomes. Okay, so S6 is a protein that makes up ribosomes. It's a component of ribosomes. Okay, and this phosphorylation that you now get of S6 ribosomes is believed to activate the ribosomes and increase their activity so that their translation ability is going to go up. Okay, so that portion of this pathway is less well understood than the other portion that I'm about to say. Okay, so that's one of the results of activation of mTORC1, that it activates the S6 kinase, which then phosphorylates the S6 protein, which is a component of ribosomes, and that then activates translation in that loose sort of way. Basically, we don't really know what exactly it does at the moment. Okay, right. Now, the other thing that mammalian targeted rapamycin complex 1 does is it phosphorylates things that are known as 4-EBPs. Okay, so let me explain what a 4-EBP is. Okay, so this is basically an elongation factor 4E, okay, elongation factor 4E binding protein. Okay, right, so let me explain what an elongation factor 4E is. So ELF4E stands for elongation, okay, that's the EL, okay. The F is then for factor, so it's elongation factor 4E. Now basically, elongation factor 4E is required for the translation of certain mRNAs. Okay, not all mRNAs, but certain mRNAs require elongation factor 4E for their translation. Okay, such mRNAs which require elongation factor 4E for their translation are known as elongation factor 4E sensitive RNAs. Okay, or sensitive mRNAs. Okay, so they cannot be translated unless the elongation factor 4E is functional, basically. Okay, now, in this set of elongation factor 4E sensitive mRNAs, you have absolutely loads of the mRNAs that are important for cell division. Okay, so important examples would be things like cyclins. Okay, which are incredibly important in coordinating the cell cycle. So basically, if you want to make cyclins, you produce the mRNA for cyclins, but you can't translate it unless you have elongation factor 4E. Okay, so it's an example of an mRNA that is an elongation factor 4E sensitive mRNA. Okay, now, basically, elongation factor 4Es are not usually available to help elongation factor 4E sensitive mRNAs being translated, okay, which usually puts a complete block on the translation of any mRNAs associated with these pro-division proteins, basically, these elongation factor 4E sensitive mRNAs. And the reason that elongation factor 4E is not usually available for helping with translation is that it's usually bound to other proteins that are known as elongation factor 4E binding proteins, i.e. our 4-EBPs. Okay, so normally 4-EBPs, and I'll get rid of that S here, are bound to the elongation factor 4Es. So here is our elongation factor 4E in red here, and here is our elongation factor 4E binding protein here. So normally the elongation factor 4E is bound to an elongation factor 4E binding protein here, and therefore it cannot help elongation factor 4E sensitive mRNAs to be translated, therefore they don't get translated and you don't produce those proteins, and that usually produces a complete block on the translation of mRNAs associated with production of proteins that are needed for cell division, such as cyclins, which are an example of an elongation factor 4E sensitive mRNA. Okay, right, so if you now want to allow the translation of these mRNAs, of these elongation factor 4E sensitive mRNAs, you need to get the 4-EBPs here to release the elongation factor 4Es. Okay, and this is one of the jobs of mammalian target of rapamycin complex 1. Basically, it can phosphorylate 4-EBPs, like so, and when they get phosphorylated by mammalian target of rapamycin complex 1, they then cleave away from the elongation factor 4Es, releasing the elongation factor 4Es, and therefore relieving this block on the translation of these mRNAs that are associated with cell division, therefore allowing the translation of these mRNAs that we are needed to make the cell divide. okay? And that's how this pathway works alongside these other pathways, such as the Ras-Raf-MEK-ERK pathway, to bring about the epigenetic change that is needed in order to make a cell divide. Okay, and that's why this pathway is so important for cancer to have found a way of overactivating, basically. Okay, so now let's talk about ways that this pathway can end up being overactivated in cancer, basically. Okay, and then what we'll move on to is the future for targeting this pathway in cancer. And nicely, conveniently for me, last year the FDA has actually approved a drug, idelalisib, okay, which is a very successful drug in treating the most common form of leukemia called chronic lymphocytic leukemia, okay, which works by blocking this pathway. It's in fact a PI3 kinase inhibitor, specifically PI3 kinase delta inhibitor. Okay, so let's firstly, however, talk about examples of how cancerous cells can overactivate this pathway so that they can overproliferate, basically. And this is going to need to be one of these very early on changes that is going to occur in the cancer, in the cells that are going on to become cancer. Okay, remember where it's usually the first genetic change that occurs to begin this progression towards cancer. Okay, right. So one of the first ways that you can overactivate this pathway is by overactivating the things which activate the PI3 kinase. Okay, so for instance, often cancer cells can have mutations in receptor tyrosine kinases, okay, RTKs. And these receptor tyrosine kinase mutations lead to the overactivation of that receptor tyrosine kinase, and then the receptor tyrosine kinase will overactivate the downstream pathways. Okay, so for instance, receptor tyrosine kinase activate the Ras-Raf-MEK-ERK pathway, which will then cause the transcriptional changes within the cell that are needed to produce the epigenetic shift that will take you into division. Okay, and then it will also activate the PI3 kinase AKT mTOR pathway, which is needed to produce the translational change that is needed for the epigenetic shift. Okay, so to give an example of where receptor tyrosine kinases have got a gain of function mutation, and this is causing the overactivation of the PI3 kinase AKT mTOR pathway, let's use the example of HER2 positive breast cancer. Okay, so there are many different types of breast cancer which are driven into the breast cancer phenotype by different things, basically. Okay, HER2 positive breast cancer is breast cancer where the key thing which causes overproliferation of the cancerous cells is that they have got a gain of function mutation in HER2, which is another receptor tyrosine kinase that binds to epidermal growth factor. So HER2 stands for the human epidermal growth factor receptor 2. So the H is for human, E is for epidermal growth factor, and then the R is for receptor. So it's the human epidermal growth factor receptor, and it's 2. Now, if you're interested, the epidermal growth factor receptor, which we talked about earlier, that is what's known as HER1. Okay, so this is another receptor for epidermal growth factor Okay, right. So in HER2 positive breast cancer, the main thing which drives the cells to divide is that you have amplifications in the gene for HER2. Okay, so you have too many HER2 genes, and this results in an overexpression of HER2 on the cells, and then they respond to the epidermal growth factor in the external fluid far too much, basically, and this drives that overproliferation. And of course, the overactivation of HER2 is then going to be overactivating both of these pathways, and that's how it leads to the overdivision of the cell. So potentially by blocking the PI3 kinase AKT mTOR pathway in these cells, we would be able to stop the cells over-dividing because of the overexpression of HER2. Okay, right. Now let's just think about, in this case of HER2-positive breast cancer, what PI3 kinase would it be that's being activated? Well, it's going to be PI3 kinase alpha, because remember, PI3 kinase alpha and PI3 kinase beta are the ones that we find in tissues other than leukocytes. Okay, and remember the PI3 kinase beta, although it bound to receptor tyrosine kinases, it wasn't particularly good at actually being activated by them. Okay, PI3 kinase alpha was the main one that could be activated by receptor tyrosine kinases, and in addition, PI3 kinase alpha was also activatable by the RAS proteins, the RAS monomeric G proteins, which were downstream of the receptor tyrosine kinase as well, whilst PI3 kinase beta couldn't be activated by RAS proteins, it required RAC1 instead. Okay, right. So there's an example of how cancer can lead to the overactivation of this pathway. In the next video we'll look at other mutations that can occur in cancerous cells which also lead to the overactivation of this pathway by different means other than just overactivation of receptor tyrosine kinases.